Thanks, Mark. And thanks to all the organizers for inviting me here today to talk to you about visualization and whether we think it's a tool or something else. So I want to start this talk by first with a discussion about the role of visualization as it plays in biology, and specifically the role it plays as a tool. As a tool, I think probably, I'm assuming people in this room would agree that visualization now has really become an important part of the data analysis workflow of biologists today. That it allows scientists to get insights about data more efficiently and more effectively, and that it turns out sometimes it can even fundamentally change the way that someone is thinking about their data and thinking about their research. And so I thought I'd start with a brief story about a tool that did just this. And it was a tool for comparing the genomes of different species, for example, the genome of a human and a lizard. So I was working with a computational biologist named Manfred Grabherr at the Broad Institute, and he was developing new computational models to compare the genomes of species. When I first started working with Manfred, he was using really standard types of visualizations like this dot plot to look at the results of his models. So what we're looking at here is one human chromosome compared against one chromosome for, in this case, the lizard. And each one of the dots inside indicated a region of sequence similarity between those two genomes. So the A, T, Cs, and Gs in those regions roughly look the same. But for Manfred to actually look across his complete data set, he had to look at many of these dot plots. One of his data sets was creating over 300 of them. When I talked to him about this, he said that he found these completely overwhelming and quite unintuitive. And it turned out in hindsight that they were hiding some really critical subtleties in his results. So we worked together for a couple of months, and we designed a new tool called MISB. And here I'm showing you a screen capture from just one of the views in that tool. And MISB was designed to support data like Manfred's, data that was complex and multidimensional. And at the time, it was the first tool to fully support this type of comparative genomics data. So what we're looking at in this view in the outer ring is all of the chromosomes from that of the human genome. And on the inner ring, all of the chromosomes from that of the lizard. The inner ring is also augmented with one user-selected chromosome from that outer ring that is then emanating a set of colored lines. And each one of those lines indicates a region of sequence similarity between the two genomes. So using MISB, scientists can quickly go through and explore their complete data set in order to try to find patterns and things that might be contained therein. So what we're looking at right here is actually a screenshot from one of the very, very first data sets that Manfred loaded into MISB. And by some measures, it's a relatively aesthetic image. There's lots of colors and there's circles, and it's shocking how much everyone loves circles. But for Manfred, what he saw was really ugly, messy data, and he wasn't expecting there to be so many lines going to so many places. And this actually went against what he knew to be biologically true. So he went back to his algorithm and he started tweaking parameters, and he was able to get but this far. At this point, he completely scrapped his model and he developed a brand new one, and that model gave him this data set. He then went on, he published his results and released his software to the scientific community. And I asked him later how long it would have taken him to make this breakthrough using the tools he had available to him prior to MISB. And what he told me was, honestly, I don't think I would ever have gotten here. So I like to tell this story because as a visualization designer, it really speaks to a success that visualization was able to disrupt and impact the way that the scientist was thinking about his research. But I want to stress that, of course, MISB is not an isolated example of having an impact on biologists in this way. And in fact, there are many, many, many tools out there today that are enabling discovery, such as some of the ones that I'm showing you up here. And these tools are actually being used by a quite large number of scientists. And so it's become just really common that we use visualization to explore biological data. But what I really want to do in this talk is I want to differentiate between the role that visualization plays as a tool for biological research from visualization design as a research endeavor itself. So visualization design research uses real-world problems in order to motivate, to ground, and to inspire the creation and testing of empirical knowledge, which in this case is knowledge about how and why visualizations work in the real world. And it's really about approaching visualization design as a science. But before I talk more specifically about what it means to conduct visualization design from a scientific perspective, I first want to address this question, which is, why in the world does it even matter to think about visualization as a science? And I want to argue for 3 points. And the first point is that your tools will be better. Some of the tools that many biologists are using today have made huge impacts, in part because there was nothing before they came along. They're sitting on top of huge data sets, or they're really combining a lot of sophisticated data management algorithms with statistical algorithms. And these are all really important reasons to use a tool. But a lot of the tools that we see could actually be improved from a visualization perspective. And I want to argue that by following good principles and structured methodologies, we can improve the tools from a visualization perspective. So the second reason that Viz as a science matters is that your confidence in your tools will be stronger. That a rigorous process, a rigorous design process, can ensure that your visualizations are showing you all of your data and only your data. And this MSB example that I just gave is an example where the previous visualization tools were in fact not showing some of the most important things going on in that scientist's data results. And the third reason is that your work will have a broader impact. Approaching visualization as a science means that not only do you get to contribute to biology by producing tools that people are going to use and make scientific discoveries with, but you can also contribute back empirical knowledge to the broader community of people who are designing visualizations more generally. And so for these 3 reasons, I'm going to focus the rest of this talk on giving you a quick overview of what it means to approach visualization design as a science, and provide some illustrative examples of what that looks like. Now, I want to note this is not meant to be a complete description of how we do this, but really just to give you a flavor for what it feels like to do this from a scientific perspective. And as a quick aside, I just want to say that defining visualization design as a research process is in fact research that I am currently doing, and I'm doing it with my colleague Jason Dykes, as well as one of my PhD students, Nina McCurdy. So many of the ideas in here are coming from all 3 of us. Okay, so if we're going to talk about science, I thought a good place to start would be to talk about validity. So validity is really, it's a cornerstone of scientific research. And what it is it means it's the extent to which a study actually measures the thing that it's meant to measure, and that it actually reflects the reality that it is meant to represent. And so when we think about validity in science, we really think about it in 2 different ways. The first type of validity is internal validity. And this is really about the truth of the cause and effect relationship. That the cause and only the cause is the thing that induced an effect in our experiments. The second kind of validity is external validity, which is the extent to which the effects that we see in an experiment can generalize to a broader population. And if you take both types of these validity into account, you end up with rigor. And rigor is really established through strong evidence for both internal and external validity. And what it does is it allows us to trust both the results that come out of our studies, as well as the generalizations that are inferred from those. Okay, so when we think about rigorous studies, I'm guessing that most people in this room are probably thinking about controlled experiments. And in these in controlled experiments, there's a number of characteristics we strive for. Replicability has become a really essential way to ensure trustworthiness of effect, that by reproducing and replicating studies and we get the same result, we have trust in those effects being true. Generalizability is also an important aspect of rigor, and this is sought through the very careful characterization and sampling of a population. Both of these characteristics fundamentally rely on the objectivity of the scientists conducting the experiments. But it turns out visualization design is not objective. In fact, it actually strives to actively be subjective. And this is because the goal of visualization design is to actually impact and disrupt the work of biologists. We want to improve their data analysis capabilities. We want to improve their understanding. And we even want to, in the best of times, disrupt the way that they're fundamentally thinking about their science. This is a very subjective process. But this lack of objectivity also implies that replicability is not possible in visualization design. And each visualization design study is actually unique, and it's shaped by all the people involved. All the scientists, all the viz designers are shaping the visualization tool that comes out, and that is not a replicable process. And it turns out that even generalizability doesn't work in the traditional sense in visualization design. How can you generalize from a really complex, unique situation where the very specific people are involved with shaping the tools that come out? How do we generalize from that? It turns out that there is a way that we generalize. It's a little different than how we think about traditional generalizability, and I'll get to that towards the end of the talk. But for now, just know generalization happens in a very different kind of way. So if we aren't objective, and we can't replicate or generalize, then how in the world do we go about ensuring rigor in visualization design work? So in the rest of this talk, what I want to do is I want to give you briefly a couple of examples of how we think about this, of how we do go about doing visualization design in a rigorous way. I'm going to do it by first talking a little bit about how we go about achieving internal validity in these studies. I'm then going to talk about some of the opportunities that we have in visualization design for strengthening the external validity of results. And then finally go back to this notion of, well, what does it mean to generalize when you're working with a very specific context and small groups of people? That's the plan for the rest of this talk. So let's dive in. Okay, so achieving internal validity. How in the world do we do this? Just a quick recap. So internal validity, again, is the degree that we are confident that the cause in an experiment produced the effect, and only that cause. And so how do we have trust that this cause-effect relationship truly exists? Well, what we do is we strive for reliability. We strive to have the same effect happen over and over again when we replicate our studies. But as I said before, again, this is not something that's available to us in visualization design. So instead, what we rely on is we rely on a notion of credibility, where credibility is about using appropriate methods in acceptable ways. And there's a growing body of work within the visualization community about how to conduct visualization design in a structured and formalized way. And much of the work that we have in this space is focused on two different kinds of models. The first one is process models, when these are models that describe the sets of things that we go through and consider in the visualization design process. And the second set of models are what we call design decision models. And these really cross-cut process models by talking about the kinds of decisions that we make along the way. And I'm going to briefly tell you about two examples, or one example for each of these. So first, thinking about process models, one of the more widely used process models today is something called the 9-stage framework. I'm showing you a pictorial representation of that here. And this framework was really designed to support people doing what's called design study research. It has a number of stages, but really one of the important bits is they're broken into three core components. The first one are the sets of things you do in the precondition phase. These are the things you do before starting a project. So for example, the very first step is called learn. And this is about learning about the space of visualization possibilities. So for example, if you're working with maybe a graph data set and you only know about node link diagrams, you're going to be severely limited in the kinds of things that you can propose because you don't know about the suite of other techniques that we have for visualizing graphs. So these are all the things you have to do before you can even start a project. The middle stages are what we call core, and these are really the main stages of a design study. And this is about going off and truly understanding the needs of the scientists you're working with, doing rapid prototyping, considering many ideas, implementing those ideas, and deploying them in the wild. And finally, at the end is the analysis stages, and this is really where we as researchers do our analytical reasoning. This is where we try to think about the lessons we've learned and the ways in which our work can generalize. And this process model also has all of these arrows, which one of my colleagues actually calls really obnoxious, which I get. But the arrows here are really meant to just depict the fact that this is a really messy, really iterative process. But still, the point here is that we can capture unique types of activities that we do along the way. And this 9-stage framework describes the kinds of considerations you may want to consider as you go through this process. So this is really just an example of one process model that we have in the Viz community that helps structure and guide the actual design process that we go through. So then I also mentioned that we have these design decision models, and the most well-known one in the Viz community is called the 9-stage framework, or I'm sorry, the nested model. And what the nested model is it presents the types of decisions we make in a nested way. So I'm going to describe what each one of these different nested layers means by illustrating it with one of my all-time favorite BioVis examples, which is the Abyss Explorer tool that was developed by Sydney Nielsen and her colleagues. So let's first talk about this outermost nested level. This is what we call problem characterization. So this is really about understanding the people you're working with, their intuitions, and the kinds of questions they have. So for the Abyss Explorer work, this was that they were working with bioinformaticians who needed to evaluate genome sequence assembly to check for their correctness. Okay, so that's the problem characterization here. The next level is what we call the abstraction. And this is about taking that problem characterization and abstracting it to a more generic way of thinking about the data types we have and the data analysis tasks we need to perform. So again, in terms of our example, this was about that these bioinformaticians needed to explore attributes of a graph in the context of the graph topology itself. Okay? This is the kind of description that we can actually design visualizations for. The next level is about the visual encodings and interaction techniques that we designed to support that abstraction. And here, Sydney and her colleagues really developed a really nice graph visualization technique that focused on being able to visualize many different attributes both along the edges and the nodes of these graphs. Finally, the innermost level is, of course, the algorithms that we have to use in order to implement our ideas. And in their paper, they discuss a variety of layout and rendering algorithms that they've optimized for this work. So one of the important things about the nested model here is not only is it just a nice way to sort of pick apart and illustrate the kinds of decisions we make, but the nesting of it really implies the fact that if you make bad decisions in those outer layers, those decisions are going to cascade inside. So for example, if I get my data abstraction wrong, it doesn't matter how brilliant my design is, I'm not going to be designing for the right problem. And so the nested model really helps to emphasize the importance of making good decisions and evaluating them. Okay, so the point here was just to say that within the Viz community, we really rely on the use of process and design decision models to help guide and frame the way that we approach visualization design. And that by doing this, we can increase the internal validity of the visualization design work that we do. This is how we achieve internal validity, is following models like these. Okay, so the next thing I wanted to talk about, opportunities for external validity. So external validity, just again, it's the extent to which the samples that we use in an experiment represent the population that they are drawn from. Now, when I say population, I actually mean this quite broadly here. Of course, populations of people, but can also be populations of data. Have we sufficiently sampled our data? As well as settings, both controlled settings as well as just a variety of diverse in the wild settings. Now, for controlled studies, I'm sure as most people in this room can appreciate, is that there's a real tension between the need to control for confounding factors in an experiment. And this really requires us to create as homogeneous a set of samples as possible. However, this often contradicts the need for representativeness, which helps us with generalizability, and which requires our samples to represent as broad a spectrum of characteristics in our population as possible. And so these are things that are often juxtaposed within controlled studies. On the other hand, designing visualizations for real people with real data, as we do in visualization design, provides opportunities to test principles and guidelines in complex real-world context, which can help us strengthen the external validity of the results by representing a much more realistic population. And I'm going to give you an example of what that looks like next. And so the point here is that there's an opportunity to use visualization design to complement a lot of the controlled work that we do. Okay, so the example I want to use is about this foundational principle we have in visualization, which is that spatial encoding is the most effective channel that we have for encoding numbers. And this encoding channel includes things like positioning marks along a common scale, like what we do in a scatter plot. And it also includes length encodings, like what we make use of when we create a bar chart. Now, we actually know that spatial encoding is the most effective from a number of controlled studies that have been done. The first of which was done by two statisticians, William Cleveland and Robert McGill, back in the 80s. And what they did is they tested a variety of participants on their ability to accurately assess values using a variety of encoding channels that included things like color and angle and spatial position. Their results indicate that the participants were significantly more accurate using spatial encodings than any of the other channels. This study was then replicated just a few years ago by Jeff Hare and Mike Bostock using Mechanical Turk. And what they got was basically they got very similar results. Spatial encodings were significantly the best. And so what we have here as an example is two controlled studies that are basically showing us the same thing, that spatial encoding is the most effective. Albeit they're doing it in rather controlled situations with rather low-level analysis tasks. All right, so a couple of years ago, I was working with a designer at the Broad Institute named Bang Wang. And what we were doing is we were trying to rethink how we might go about visualizing gene expression data, how to move away from the more traditional heat map way of looking at them. At the time, we were working with a group at the Broad that was studying yeast. And this group had been collecting gene expression information about multiple species of yeast, and they were doing it at different points in the life cycle of the yeast. But what they were really doing is they were struggling to make sense of nuances in their data using the typical heat map. And so Bang and I decided that we wanted to explore how we might move away from color and to instead use the spatial encoding channel. And so we experimented with a variety of different ways that we could imagine showing their data spatially, which ultimately led to the development of a new visualization technique that we called the curve map. Now, when we gave this technique to the yeast scientists, they were able to confirm both things that they knew already in their data, but then they started seeing things that they had never seen before. And this led them to have new hypotheses and ultimately to design and run brand new experiments. And so we use this result as validation that this basic principle of spatial encoding, being better than color, held up in this real-world scenario involving much more complex and ecologically valid analysis tasks. About the same time that Bang and I introduced this curve map idea to the group working with yeast, I was also working with a group at Harvard Medical School that was studying gene expression, but this time in fruit flies. They also were relying on heat maps to make sense of their data and agreed that their fine-scale patterns were really, really difficult to discern in this visualization. So I suggested to them, hey, let's try this new technique called a curve map. And initially they were resistant. They said things to me like, well, can we put a button on the tool that'll switch back and forth between the heat map and the curve map? It's fine, okay, we'll do that. But over time, as they started using the curve map, they really warmed up to the idea. And at the end, the final tool that we designed, shown here, ultimately did not use heat maps at all and only used the curve map. Okay, so even though this is let me say one more thing. So then the group, after they did analysis using this tool, they made some discoveries. They started writing up their paper. And it turns out that in their published paper that they used the curve map to show gene expression data instead of the more traditional heat map. And more specifically, they were using these overlay charts at the bottom in their figures in their paper in order to illustrate how the gene expression varied in different parts of the data set. So this is, again, another sort of example of a validation in a different real-world context of this basic principle. But I think for me, what was probably the most important bit of validation we got was made by one of the senior authors on this paper, Michael Eisen, sometimes known as the father of the clustered heat map for showing gene expression data. And his response when he saw a draft of this paper was, yeah, that's the right way to show this data. So for us, that was a really strong sense of success and validation that, in fact, this is a good technique for looking at this kind of data. So what I showed you here is about this notion that we have this foundational principle, spatial encoding is most effective. It was something that was established in the community through a number of controlled studies. But then also I've shown you 2 examples of using this in the wild with real people, real data, real problems that strengthens the external validity that this is in fact a good principle and one that is valid and rigorously tested. And so visualization design, what I want to argue to you is visualization design gives you many opportunities to test existing principles and guidelines in the wild in order to strengthen the validity of our results. And this is something I think is a huge missed opportunity. Okay, so then the last bit of the story that I wanted to tell you about was how generalizations emerge from visualization design. How can we end up making broader claims for a process that is inherently context-specific, bespoke, and complicated by bias and subjectivity? Like, how in the world does that happen? So I feel like I just keep dangling this in front of you. We'll get there in a second. So first, generalizations. What do generalizations mean? So generalizations are claims that infer effects that are seen in an experiment beyond the specific samples to a population at large. That's what a generalization is. Now, for us to have trust in these generalizations, studies need to have both strong internal and external validity. Generally speaking, however, it's pretty difficult to have both kinds of validity be really strong in any single study. So instead, what we rely on quite often is this notion of combining complementary studies, and I just gave you an example of how we did that for that basic visualization principle. So we can combine complementary controlled studies with more applied work to strengthen the external validity and to come up with a good chance of having trustworthy generalizations. And this combination of controlled and applied work in some academic communities is referred to as mixed methods. Now, this combination of studies, however, seems to infer that testing basic principles and guidelines is the thing that we should be doing in visualization design. But is that all that we can do in visualization design? I'll answer that question for you. The answer is no. It turns out, though, in working through the complexities of designing a visualization, and anyone in this room who has gone through this, you know that there are many challenges that you face along the way and that you learn a lot through that process. That there's things like how do we establish good collaborations with our collaborators? How do we effectively elicit feedback from the people that we're designing for? And then there's all the unexpected uses of a tool that someone will do that the tool was never meant to support. These are all things that, important insights that we acquire through the visualization design process that are of potential broader interest to others who are designing visualizations. And so this is, in fact, a type of knowledge that should generalize, but it generalizes in a way that's typically different than what we think of. And it does so through a process of transferring knowledge to a new population. So this notion of transfer is about inferring effects from one population to another. Now, the way that we do this is by making judgments on the similarity of the two populations. And then the other thing that's worth noting here is that transfer is a type of generalization that isn't done by the people conducting the initial study. It's actually done by an independent researcher. And this is because those original researchers can't be expected to know the entire space of all the other types of populations for which, in effect, might transfer for. Instead, it is the independent researcher who is working on those problems that can decide whether or not there's a similarity between the populations and that, in effect, could potentially transfer. Okay, so that's one of the things that makes this really different than the way that we typically think about generalization. And so I'm going to give you 2 examples of transfer just to give you a flavor of what this looks like in practice. So one of them, in fact, we've already seen, okay? So this example about working with a group that looked at gene expression in yeast and a different group that was looking at it in fruit flies. It was about transferring this new technique, this curve map, from one domain to another. And in this case, we were able to make the judgments about the similarity of the structure of the data, as well as the kind of tasks that both groups of scientists wanted to be able to do. So based on these similarities, we proposed the curve map to the second group, and in fact, we showed that it worked. And so this was a type of transfer that was very successful. But to give you a slightly different example, I want to talk to you about a transfer that we did within my group for a technique called creativity workshops. And this was a method that was first proposed in the VIS community by the group up here, GI Center in London. And they originally reported on this by running creativity workshops with energy analysts in the UK. So creativity workshops are a very, very cool method. And what they're really good for is very quickly and effectively being able to elicit design requirements through structured brainstorming as a group. And so this paper was the first to bring those up in the VIS context. Well, so one of my PhD students got a hold of this paper, and he got really interested in trying out this method within a new collaboration that we had with some neuroscientists at Utah. He got excited because the challenges that he was facing working with this neuroscience lab seemed very, very familiar to some of the challenges that they describe in this paper. Challenges that they claim that the creativity methods, creativity workshop really helped to overcome. And so he had hoped that these creativity workshops might be effective for jump-starting his own design process with these neuroscientists. So based on the description of the method in the paper, we designed and conducted a creativity workshop with these neuroscientists. Now, this workshop ended up leading to a number of cross-cutting ideas. These ideas led to several visualization tools and publications in both neuroscience and visualization journals. And so the punchline here is, yes, this method did in fact transfer, and it transferred beautifully. In fact, it transferred so beautifully that this is a method that we're using in almost all of my visualization design projects now that we conduct within my research group. And so this is really just another example that shows that transfer doesn't have to just be about transferring specific visualization techniques. It can also be about transferring the types of methods or the things that we do throughout the design process as well. But there's this question about, well, how do we go about supporting transfer? And again, reminding you that transfer is something that has to happen externally. It's an independent researcher that does it. And it really requires on detailed descriptions of process, people, and context. Now, what does it mean to be detailed enough? This is actually an open question in the visualization community and one that several of us are still thinking about and tackling. But it's really important to be able to describe the details of the process you went through and the people involved in that in order for someone else to determine, one, if there's a similarity between those two populations, and two, also why we think that that type of method or technique might be effective in this other space. Okay, so this was really just to give you a flavor that generalizations can and do in fact emerge from visualization design itself, but they do so in a slightly different way than what we're used to thinking about. So just going back to this initial question, so how do we ensure rigor in visualization design? I'm hoping that in this talk I've given you a little bit of a sense to know that this is in fact possible, that we can do through by ensuring internal validity through the use of appropriate and established design methods and methodologies, that we can also do this by increasing external validity through having diverse sets of scenarios in which we test our principles in, and also that we can generalize through this notion of transferring knowledge, but that this really requires us to have detailed, rich, and thick descriptions of the design process itself. And I hope you will at least give me the benefit of the doubt of considering that if you do in fact think about these things, that your tools can be better, that you can have more confidence in them, and that your work has the potential to have a much broader impact. And so with that, I just want to say thank you all for your attention, and I'm happy to take any questions you might have.
Okay, thank you, Miriah, for the super interesting talk. Questions?
Yeah, could you describe a little bit more about how the creativity workshops actually work?
Great question. So I wish I had a paper to point you to. in 6 months, I will. But these are workshops that rely on a lot of research in group creativity. So how do you bring people together, make them creative to support brainstorming? And we do it through a series of activities that are first divergent to produce lots of ideas, then followed by convergent activities to hone in on ones. So we do things like, for example, one of the activities is what's called a visualization awareness activity, where we present different types of visualizations that have nothing to do with the problem that the group is working on, to get people just thinking about the space of possibilities and to get people talking about it. We then do things, like asking them questions like, what do you want to see? What do you want to be able to do? What are your challenges? If that challenge was gone, what would you be able to do next? And so we have structured activities that go through those sorts of things. I'd encourage you to look at that paper that I referenced. They give some fairly brief descriptions of it, but we are in fact working, my PhD student is working on a paper that really goes into the theory behind.
So the Graffinity paper is the one that you referenced?
This one, with energy analysts.
Oh, okay.
You can send me an email and I'd be happy to.
I wrote that one down. Thank you.
Yep.
Next question.
Hi, your talk made a lot of sense. Could you go back one slide? I said did make a lot of sense.
This one?
Yeah. Yeah, and for number one, how are you going to decide which design methods to use? Presumably people have proposed different design methods. How do you know which ones are better than others? That seems to me that that's part of the rigor here.
Yep. Well, the first question I would ask is, like, how do you know in a lab setting which types of statistical analysis or even wet lab experimental procedures are the best ones to use? And I think the answer in design is the same one, is that it's kind of based upon standards that the community over time decides has been successful. And so you know, this 9-stage framework I presented is one that now has, in the 5 years the paper has been out, it has almost 300 citations of people using it. And so that is, of course, an imperfect and subjective measure, but it's one measure that shows that people are using this and they're finding it helpful to get to successful visualization designs. The nested model is another example that's highly cited and people talk about all the time. So it's something that as a community we've decided is useful and impactful. And I don't think that that's all that different than what happens in controlled studies sometimes as well, like why you choose to use a specific technique over another. Some of it is experience and some of it is just test of time.
It seems like there should be some more objective way of evaluating different design methods.
Do you have any ideas?
I'll have to think about it.
Okay. Yeah, honestly, this is ongoing research. So if you have ideas for these sorts of things, I would love to hear them. I really would.
So I was wondering if, so the curve map you invented, there's like 5,000 genes, so why are there not like 5,000 plots? Like, how do you merge them?
Great question. You picked up on that. Yeah. So the curve map is not without problems, and in fact, I think one of the reasons people really like heat maps is it's what we call it's a very data-dense display. You can get many, many, many points into a single display, and obviously with the curve map, you can't show the same amount of data. However, I'd argue that probably what you do with that much data is different than what you do if you're looking at a smaller set. When I've talked to people about how they use heat maps, they say, well, I get some general overview that there's roughly these classes of things. But making really fine-scale distinctions between, you know, is there variations in time? Are things shifted? They're not using heat maps for that kind of thing. So then the curve map is really something that doesn't scale in the same way a heat map does, but I would argue is going to support a different set of tasks.
Yeah, but how are they? Like, how do you decide whether there's 8 or there's 11, and like how are they? Like,
how do you decide which ones to look at?
Yeah.
In both of these projects, like for example, with the fruit flies, they were looking at roughly 50 genes. And in the yeast, it was on the order of a couple of dozen. And part of what we designed, you know, so the curve map was a technique that was part of an interactive tool. So for example, with the yeast group, a big part of the interactivity was giving them mechanisms to support which genes they wanted to look at and explore to populate the curve map.
Okay.
It's a good question, though.
So what role or weighting do you give to aesthetics and art in what you do?
Oh, such a great question. I really want to think about this deeply. So I think this notion of aesthetics is a really interesting one, like even trying to define what that means. You know, if something looks good and feels good, is it because we've somehow made it pretty? Or is it just because we've really nailed the functionality and the basic principles? And I think that's kind of an open question. And I think there's also this other really interesting, not completely related, but this question about trust and branding. So I've talked with other viz designers and noticed things in the world where we see certain, to the general public, certain data is presented in certain kinds of ways, whether makes you want to think that it's playful or scientific or these sorts of things. And I think that aesthetics plays a really important role in some of the decisions we make based upon the messaging. You know, we like to think that we're unbiased and objective, but in fact, we're trying to message that I'm a rigorous scientist who's giving you unbiased data. I'm probably not going to put like flowers and lots of colors around it. I'm going to make it look as sort of bleak and minimal as possible. And so I think this notion of aesthetics is such an interesting but really complex topic that in the viz community people haven't really looked at yet. And I think there's a lot of room to think about that.
So for you, is it all functional then? So no art, no room for art. It's all about the function of the visualization.
I think there's a lot of room for design. You know, and I think again, I think it comes down to what your goal is. Like, you can go to a museum and you can see what some people might call a visualization and other people might call data-driven art. And I think there's a lot of room for that. But I think when it comes to the, you know, especially for an audience like this, creating exploratory tools that's really to support analysis, that there's a lot more overlap between this kind of work and the goals that designers have when it comes to aesthetic, more so than when it comes to artists. But that's not to say that there aren't things about art that are potentially really interesting and important for certain kinds of visualizations.
Thank you. Neil?
Great. Excellent talk, Miriah. I really enjoyed it. I'm very curious if you can give us here in the room some practical advice, rather than talking about art and stuff like that. I'm curious, so what would you, based on your research, suggest for how should we evaluate BioVis papers? Like, how should we figure out whether the proposed techniques are good or not?
It's a great question.
Sorry, I should have
I feel really on the spot. You know, I think the sort of standards that have emerged within the viz community for looking at applied work, which is something that is still evolving and is still relatively new even with that community. We struggle as well there about how do I evaluate this, you know, visualization design study. And I think what I would argue for that it's really about looking at, you know, did someone just throw spaghetti on the wall? Or did someone go through some sort of structured process that really considered the design, considered the decisions they were made, and can justify why you make certain decisions? Did someone really talk about the data in a fairly generic way that I think is also important for justifying why you make certain decisions or the others? And I think, you know, in my own reading of BioVis papers, I think that's one of the things I see lacking a lot is a description of, well, how did you actually get here? And can I trust that what you're showing me is good? And a big part of that trust comes from knowing what you did and seeing that it was a structured process. That's one of the things I think could be really improved, both here as well as in the viz community.
Great. Thank you very much, and sorry for putting you on the spot.
We have time for one last question.
Sorry. Yeah, great talk. I was wondering if you could elaborate a little bit more on the interaction in your designs. Like, part of what you've shown is basically a decision to reduce the overview or the data intensity into a more iterative process of selecting data and, yeah.
Yeah, so all of the visualizations that I've shown you are part of interactive tools. Interactivity, I think, is something that we just can't do without, especially as data set sizes get even remotely large. And I think this notion of very data-dense visualizations just in practice doesn't work very well. And so instead, what's really useful is this idea of how do we give someone guided exploration through a tool, right? How do we give them a window into the data that lets them select things of interest that we can then see things in more detail? And that's sort of the basic principle that I use all the time. An interesting note about interaction, though, is that interaction is something that's not studied very much in the viz community. There's a whole other part of computer science called human-computer interaction where they study that more. And it is so fundamental to the way that we design exploratory tools, and yet we don't know a lot about the sort of theory and basics behind how and why it works within the viz community. And so, you know, it's a little bit less rigorous the way that we go about designing interactions, but it is something that you'll see, and you'll see very few static tools anymore, just because any data that's of interest is too big for that.
Okay, thank you very much.